All right, I believe we are live again. Okay, so I hope you guys enjoyed that series of presentations from today. There will be more coming soon, but in the meantime, we have a bit of an interlude, I suppose. Uh, so, for those of you who aren't aware, which is probably most of you, uh, Incidents and Reflections was a radio show slash podcast that I used to co-host when I was a geology undergrad at the University of Maryland, and it was created by my fellow geology major Alex Jacob, who later transferred to Colorado State University, but uh, he brought me on as a co-host, and I continued it after he left, and later on, my good friend Joan Turmelle, um, who went to East Carolina State, is that right?
East Carolina University,
East
yes.
Carolina University, yeah, sorry. Um, she was studying anthropology at the time. She offered to be my new co-host, and she would Skype in every week, and we would do the show together. And so, as part of this fundraiser, I guess we have decided to bring it back just this one time, and we will be talking about some of the interesting science news, which was what we talked about back then, that caught our eye in uh, the past few months, I guess. Um, so feel free to send in your questions to us, either about the stories we cover or about our areas of expertise. So in my case, it would be like bird evolution. In Joan's case, it would be anthropology. Um, and yeah, I suppose we can get started. So when we did this, WMUC, in case you're wondering, was the local kind of radio station of the University of Maryland. Of course, we're not doing this in collaboration with them right now, but it was kind of just there on our old logo. So that's what it is. Um, so usually before we would dive into kind of the news items in terms of like research papers, we would often do kind of promotions of like science-related media that we came across, like either recently released or about to be released. So we're kind of gonna do that again here. So I guess we can start. I'm gonna go to the next slide, and we can start off with a prehistoric road trip with Joan. I'm sure will be happy to introduce.
Yes, so this is a brand new show. I'm sure a lot of you are familiar with it by now, but if not, this aired on PBS last week on Wednesday. This is a show by Emily Grassley. If you're not familiar with her, she's a science communicator who's currently based at the Field Museum in Chicago. She runs the very popular natural history themed YouTube show, The Brain Scoop, which premiered in 2013, which runs a whole gamut of different conversations between her and colleagues with different themes, ranging from zoology to botany to anthropology. And so this has been a dream of hers for quite a while, and she was able to get it finished just in time for everybody at home who's certainly going to be able to watch it since
Right.
TVs will not be far from them, certainly. Um, the basic format of the show is that Emily Grassley is going towards her hometown in the Midwest United States, visiting different fossil sites throughout the region, and kind of giving a overview not only of the field of paleontology, but of the deep time of the Midwest United States. And so this show has already aired, as I've mentioned. Last Wednesday, they had the first episode. Um, and just passing thoughts on it, it was very well done. They used a lot of graphics that explain rather complex science concepts,
Yeah.
and they introduced a lot of fascinating individuals and really cool sites. One of my favorites in particular was the Bear Gulch site in Montana. This is
Mm.
a Carboniferous site where a lot of very bizarre fossil fishes had been found. Um, and what's really funny is like throughout the show, there were little tiny tidbits about what field paleontologists use to find their remains, and they were mentioning that the oil from sharks' livers gives off a very potent smell,
Wow.
and so you can kind of smell it in the rocks, and you can know that there's a shark somewhere near here. And I thought that was really funny and unique. Um, but
That's
if you
amazing.
happen to want to catch it, I believe it's been uh, put on their website, a PBS website for streaming.
Yeah.
Otherwise, of course, you can buy the DVD and whatnot. And this Wednesday at 10 p.m. Eastern Standard Time, the second episode is going to air. So I hope everybody gets a chance to catch up with that.
Excellent. Yeah, yeah. So there are going to be 3 episodes in total. Is that it?
That's right. Yep.
Yep. Yeah. So do catch those upcoming episodes and watch watch the first one if you can. Yeah, unfortunately, we don't get PBS here, though. I'm currently in the UK, as you might have noticed. I cannot access it at the moment, but I definitely look forward to seeing it myself. And do you have anything else to add?
Nope, you can go right ahead.
All right. So let's see. Next item. Okay, so I'm quite interested in kind of scientific outreach, and I'm always looking for interesting ways of doing scientific outreach. Or, not necessarily myself doing it, but ways that other people have come up with doing it. And one of the ways that I've quite enjoyed experiencing science outreach, I guess, is through music. And so I listen to a lot of kind of science-related songs with science-related lyrics, things like that. Um, so John Hinton is a British performer and science communicator who specializes in writing science songs. He has written a series of shows, live shows that he plays on stage called the Ensonglopedia series. And in each of these shows, he has 26 songs, one for each letter of the alphabet about a certain theme. So one of the shows I've seen him present is the Ensonglopedia of Animals, which is 26 songs about 26 different types of animals, and not always like well-known animals you'd find in like in a kid's alphabet book either. But he goes through like not only mammals but also other types of vertebrates. He has I think 3 different songs about fish. He has leafy sea dragon and wobbegong shark and lamprey, and he also goes through various groups of invertebrates as well. So he has songs about sea slugs and leeches and several kinds of arthropods, water bears, nematodes, and even hydras and sponges. So it's a very interesting show, and he actually arranges those phylogenetically in terms of how distantly related they are to humans. So he starts out with the first song about humans and goes on to our more distant relatives. Um, and so his latest project is called the Ensonglopedia of the Human. And so, thank you. This is going to be 26 songs about what it means to be human. So, as you might see from the image on here, he actually gave a kind of live streaming performance of the show about a month ago in May. But it is still a work in progress. So he has only written about half the songs that are going to be in the show, I believe. And so he kind of gave a sample of those during his live stream. And the live stream is, as last I checked, still available to watch on his Facebook page, which you can see on here. If that sounds interesting to you, do check it out, and please give him some feedback on what you think about the songs and what topics you think would be worth covering in a show like this.
Yeah, unfortunately, I haven't had the chance to check it out myself, but I definitely should very soon.
I would appreciate your thoughts on it, because after all, it is a very anthropology focused.
Oh, absolutely. Because I did see like a little peek at the song list.
Right.
And it looked like he wasn't just covering. He was covering like cultural anthropology as well as biological anthropology.
Yeah, he covers both aspects in the show. So he has songs about like different aspects of both of those fields, I guess. Which is pretty cool. And if you have some ideas for other songs he could include, I'm sure he would be happy to hear it.
Yes, absolutely.
Okay, do you want to talk about Light Through the Ages?
Yes, absolutely. So this is a brand new book that just came out a couple months ago. I want to say in March, and it's by a paleontologist and paleo artist named Mark Whitten. Mark is well known for his reconstructions of pterosaurs. In fact, he published quite a few years ago like a landmark book on pterosaur research and evolution biology. It's certain to become like a frame of reference for a lot of budding pterosaurologists. For this particular work, he is directly inspired by a book from the 1940s called Life Through the Ages. So this is the first one. And this is by a very little-known paleo artist. I'm not sure a lot of you heard of him, Charles Knight, who was very big in the late 1900s, early 20th century. He was a foremost authority on mammal anatomy, and he reconstructed many prehistoric scenes, including dinosaurs that have appeared in many museums around the world, including the American Museum of Natural History and the Field Museum in Chicago, where they are still on display. And what Mark Whitten did was he took that concept, which is basically a run through the history of life with comparisons with modern species. So, like, for example, Charles Knight would look at fossil elephants, and then he would examine living elephants. Incidentally, with these beautiful charcoal drawings in the book, Mark took that and expanded upon it and updated it for the 21st century. So what you have here is a series of plates by Mark, full color, beautiful illustrations, going through the whole history of life from the formation of the Earth to the Anthropocene event, and he kind of gives a rundown of the sequence of events that occurred. So I would argue, like, if you're sort of a budding paleontologist or interested in a brief overview of what the state of the science is, this would be a good reference to check out. So like it's good not just for the art, but for the text as well.
Mm-hmm.
Um, like I thought it was fairly well balanced. He spends a lot of time with invertebrates and floras and just basic aspects of ecology, as well as with dinosaurs and big mammals. So it's a
Right.
well-rounded collection of information and beautiful imagery. I was particularly taken by his Pelagornis, incidentally,
Oh yeah.
Pelagornid that he made in the book. There's cameos by recent discoveries as well. So the Ankylosaur Zool
Yep.
shows up in this book as well, literally described as the destroyer of shins, which I thought was particularly hilarious.
That's what it
Um,
is. Yep.
and as you can see on the website, we have the link to the book in case you're interested in purchasing it. But of course, it's available in general bookstores, Barnes and Noble, Amazon, and so forth. But I highly recommend.
Right. Excellent. Yeah, I definitely look forward to getting a copy of my own. It sounds fantastic. All right. Do you have anything else to say?
Nope, you can go right ahead.
Alrighty. Okay. I guess our kind of our last promotional item is going to be the PhyloCode. So a bit of background, I suppose, is that vertebrate paleontologists have, in general, largely shifted to a system of naming groups based on phylogenetic relationships, so how organisms are related to each other. So they will define a certain group as being like anything that's more closely related to species A than to species B is a member of a given group. Uh, and there are a few variations on that type of definition, but that's the general idea. So instead of grouping organisms together based on potentially arbitrary kind of physical features or things that can easily get lost or regained in evolution, they're grouped based on like their actual evolutionary history and relationships. Um, and we've been using this system for a while, and um, have shifted over to using it. And for most part, I think we've gotten pretty used to doing it. But there hasn't really been any kind of formal regulation of what definitions are valid. If two people name the same clade different definitions, um, which one should you use, that kind of thing, or what definitions go with what names. There hasn't really been any kind of formal kind of regulation of that, unlike the ICZN, which is a kind of International Commission on Zoological Nomenclature for Linnaean taxonomy, at least up to the family group level. For a long time, this has been in preparation. This PhyloCode has been in preparation to serve this kind of role to regulate what definitions take priority and how you should define groups. It has been in the works for a long time, more than a decade, probably more than 2 decades. And finally, it is now out because the companion volume called Phylonyms, which is basically a compendium of select groups that have been defined following the rules of the PhyloCode, has been released. And basically, the PhyloCode is designed to go into effect at the same time as Phylonyms gets released. And so Phylonyms got published, I think, a couple months ago. So now PhyloCode is finally valid and can be used. And they also have an online database called RegNum. Um, RegNum, which kind of records all of these definitions that have been established for these groups. Now, since we've gone for so long without really any this kind of regulation, I feel like a lot of the community are kind of ambivalent about PhyloCode. Like they might not be that interested in actually following its regulations because we've gone so long without it. What's the point? And I know some people have raised problems with how some of the groups are defined in phylonyms because they don't always follow like exactly the kind of historically commonly used concepts of some of those names. Um, and and so on. So it remains to be seen whether how influential this PhyloCode is actually going to be. But at least it's there. It's finally out, and we've been waiting for it for a very long time. Um, all right. So I guess before we move on, we did receive a question before the show started, didn't we?
Yes, I think so.
Let's see. So this question comes from Joan's sister, Julie. Is that right?
Is that
Yes.
right?
So she says she is very proud of you for using your amazing talents to fight for positive change in the world. That is sweet.
So sweet.
And her question is, if dinosaurs lived in outer space, how would they survive and thrive?
Ooh, survive and thrive. Well, I guess that question's sort of tied to human exploration of space.
Right.
As living organisms, there's a certain set of criteria that we need in order to function. Mentioned, stable sources of water and the right sort of atmosphere to breathe, plenty of resources to use, food sources. I don't see, humans really getting much out in space without, extensive technological aid,
Right.
let alone something like a dinosaur.
Right. Right.
I
Yes.
can't even think of if any birds have been brought into space the way that dogs and chimpanzees have been.
I think chickens have, or chicken embryos have some. Yeah, I don't really follow it that closely either, but
Okay.
but yeah, yes, yeah. I think that's the gist of what I would say too. Like, probably not without like extreme technological assistance. Um, and as far as the dinosaurs that are around today, or even the Mesozoic ones are concerned, that technological assistance probably wouldn't come without assistance from another species like humans.
Oh, absolutely.
Yeah,
So,
but
don't expect Jurassic Park and don't
in
expect
space.
Space Jurassic Park anytime soon.
Yes, I guess hypothetically, if we had like sapient dinosaurs, they could develop the means to get up into space. But for now, I guess it's a long stretch to say the least.
Sure, it's an open question. Let's just
Yeah,
say that.
yeah, for sure. Okay, so I guess we can go on to our like research news items after this, which will take up the bulk of the show. So for the first news item, I had Asteriornis here, but you've already heard a lot about Asteriornis from my lab mate Juan and my supervisor Daniel. I don't think I could add too much to what they said. So basically, yes, Asteriornis is a very cool find, kind of the best preserved skull of any modern bird. In fact, one of the best preserved skulls of any kind of bird in the fossil record. Um, and it's probably a close relative to the last common ancestor of chickens and ducks, showing us what this group of birds was kind of like in the Cretaceous. And again, like Daniel said, it provides support for a lot of the things we predicted we would find in latest Cretaceous modern birds, quote unquote modern birds, because obviously they weren't literally living in modern times, but belonging to the crown bird group. So I guess that I shouldn't blather on too much about Asteriornis, so we can move on. Right, so we have each picked kind of two what we think are interesting studies to talk about. So this is, I guess, my first contribution. Um, and this is about the ontogenetic dietary strips of Deinonychus. And a Deinonychus, of course, is a very well-known dromaeosaur dinosaur, quote unquote raptor dinosaur. Um, it is well known for various reasons, including kind of kickstarting the dinosaur renaissance. It was discovered and described by John Ostrom in the 1960s, and from there he kind of revived the idea based on similarities between Deinonychus and Archaeopteryx that maybe birds had evolved from dinosaurs, and that and also based on the anatomy of Deinonychus, that maybe some dinosaurs, at least some Mesozoic dinosaurs, were kind of highly active and potentially metabolically had elevated metabolisms compared to your standard reptile, um, and both of those conclusions are, quite widely supported today. So Deinonychus has been kind of scientifically, historically important for that reason, and of course, it is also well known, possibly for being a likely basis for the quote-unquote raptors in Jurassic Park, which were given the name Velociraptor, but the actual Velociraptor was a much smaller animal than Deinonychus, which was about the size of a wolf. Um, yeah, because at the time Michael Crichton was consulting Greg Paul's Predatory Dinosaurs of the World, and Greg Paul considered Deinonychus to be a species of Velociraptor, which no one else really uses that taxonomy, but that's what he had to go on. Um, yeah, so Deinonychus is also well known for, John Ostrom actually suggested this, that it was a pack hunting animal because um, kind of we sometimes find numerous specimens of Deinonychus found associated with a plant-eating dinosaur called Tenontosaurus. And in fact, it's kind of a meme at this point that Tenontosaurus, whenever it's shown in any kind of media, it's always shown being attacked and eaten by groups of Deinonychus. Um, yeah, there's just no other reason people show it in popular media. The only reason it's shown is to be Deinonychus food, which is kind of unfortunate.
Walking buffet.
Yeah, yeah, because it's actually a pretty well known and interesting animal in its own right in terms of its anatomy, but yes, that is its claim to fame, I suppose. Um, but the question of whether Deinonychus really hunted in packs is a somewhat controversial one in paleontology because there are actually several ways one could interpret the evidence that we found. Maybe this was kind of a chance gathering of several Deinonychus that came together to scavenge on a Tenontosaurus carcass. Maybe only one or two of them brought the Tenontosaurus down, and the others came when they saw that there was a meal available and they were fighting over the scraps, things like that. It's actually kind of ambiguous whether this evidence really indicates that they hunted in packs. Um, and so a new study kind of tried a new approach to this problem. What it did was it looked at the isotopes in Deinonychus teeth, as well as the teeth of some other animals that lived alongside. Um, and if you caught David Fox's talk yesterday during the stream, um, he talked about basically isotope, he's applying isotope geochemistry to study ancient ecosystems. And I'm not a isotope geochemist, so I can't really, I'm not that well read on the details. But basically, it operates on the principle that you are what you eat. Basically, what you eat gets incorporated into your body tissues, and so animals feeding on different diets are going to have different proportions of different isotopes incorporated into their teeth and bones and other tissues as well. Um, and so what they did was they looked at carbon isotopes in the teeth of different age classes of Deinonychus, of juveniles, subadults, adults, as well as different age classes of goniopholidids, which are close relatives of crocodilians that lived at the same time as Deinonychus. Um, and they also looked at these guys across different age ranges. And what they found was that juvenile goniopholidids um, tended to have a kind of what they say are more enriched carbon 13, are more enriched in carbon 13 than the teeth of adults. And what this usually reflects is that higher enrichment in carbon 13 means that they're feeding higher at higher trophic levels, so higher in the food chain, which is interesting because these are juveniles, and you might be thinking that doesn't make a lot of sense. Why would juveniles be feeding higher in the food chain than the adults? But what happens is that we see this in modern crocodilians too, is that the juveniles often are going after very small prey like insects and spiders and frogs and things like that. And those small things, a lot of them are not like herbivores. They're hunting other insects or other spiders, or they're hunting invertebrates, things like that. So in this event, the juvenile crocodilians are actually feeding several levels up the food chain by hunting these small prey items, whereas the adults might go after a kind of broader spectrum of prey. So they will be hunting things like fish. They will be hunting some types of land animals, perhaps, and some of those animals are going to be primarily herbivorous animals. And so in this way, the adults are often going to be feeding lower on the trophic level than the juveniles are. And so this is seen in modern crocodilians, and they also found this pattern in the goniopholidids. Yeah, a bit of a tongue twister there. And so when they applied this method to Deinonychus that was found in the same localities as the goniopholidids, they found that the juvenile Deinonychus showed a very similar pattern. They were greater enriched in carbon-13 in their teeth than in the adults, and so that also suggests they were feeding on different prey items from the adults. So they used this to argue that perhaps Deinonychus didn't really have a social structure similar to what we might see in a mammalian pack hunter today. So something like a lion or a wolf, probably not something like that, because in those species, the adults go out and hunt, and they might bring down a herbivorous kind of prey item, and then they will either bring some of it back to feed the young, or have the young come in and feed on the carcass. Um, and so basically, in this case, the juveniles would be feeding on the same kind of prey item as the adults are. But in this case, it seems like the juvenile Deinonychus were were doing their own thing, and the adults were doing something else. So the adults probably feeding on herbivorous dinosaurs, whereas the juveniles perhaps feeding on insects or small reptiles, small amphibians, small mammals, things like that. So does this indicate that Deinonychus was not a pack hunter? Well, I would say not exactly. There are definitely a lot there's definitely a lot of variations that could such behavior could potentially take. So it could have been that the adults were hunting in packs and bringing down larger herbivorous dinosaurs, whereas the juveniles were off doing their own thing and just hunting small prey on their own, and just doing something different, which would be consistent with what we see in terms of that niche partitioning, is consistent with what we see in a lot of large carnivorous reptiles today. Um, and so, well, um, it is a very interesting approach to answer this problem, and I think it sheds some real light on the biology of Deinonychus. But I do think the question of sociality remains open. But yeah, definitely, I don't think I do think that this indicates that they didn't exactly behave like kind of a mammalian pack hunter would. So that is quite interesting. Do you have
It's a
anything?
fascinating paper.
Yeah,
I'm curious if this has been done with other dinosaurs where we know the identities of the adults and the juveniles, or if
right.
that's something that's planning on happening.
Right. Yeah, it would be interesting to see this applied to other types of dinosaurs. So isotope kind of studies have been done on other dinosaurs before. So, for example, there is one where they use it to study whether or not spinosaurids might have been feeding from aquatic environments, and in
Oh,
that
of
case,
course.
the answer was yes, they probably were, which is consistent with other lines of evidence, of course. There was one where they used this to suggest that Camarasaurus, which is a Jurassic sauropod, was migrating because they showed that it incorporated different isotopic signatures into its teeth during different times during the course of its life on what seems to be I think an annual cycle. So that is also really cool. So you can find out a lot about the biology of extinct animals from isotopes. Yeah. Do you have anything else to add, June?
Nothing to recommend.
All right, so I guess my second story on here is going to be about also about kind of dinosaur biology. In this case, kind of thermophysiology, which is a hot topic to say the least in dinosaur paleontology. So this is a very interesting study by Lovelace et al. And also Scott Hartman, who you might have heard from yesterday, was one of the co-authors. Um, and so what they did was they used a program called Niche Mapper. So Niche Mapper is a program where you can enter parameters about environmental conditions. So what the climate was like, what the temperatures were like, what the wind speed was like, how humid it was, all kinds of things. And then you can model an animal. So you specify its body shape, you specify its metabolism, you specify, you can even specify things like behavior, like whether or not it lives in burrows, things like that. And then you can use that to study, whether or not a given environment is a suitable habitat for a certain species that you're looking at. Mostly this has been used to study living animals, so things like a lot of the studies on like how climate change is going to impact the ongoing anthropogenic global warming is going to impact different types of animals has been done using niche mappers of, will this habitat still be suitable for species in 20 years, that kind of thing. Um, but this study decided to apply this method to some extinct animals, so Triassic dinosaurs. And so what they did was based on previous paleoclimate studies, they kind of specified the conditions of climatic conditions of Earth during the Triassic. And then they modeled two types of well-known Triassic dinosaurs. One of these was the earliest theropod, a small theropod, like smaller than a human on average, Coelophysis. Um, And the other one was an early sauropodomorph, a much larger animal. This is Plateosaurus, so something the size of a horse or a rhino. Um, and both of these are known from very complete skeletons, are well studied. And so they modeled these two types of animals and threw them into the kind of the Triassic world that they modeled within Niche Mapper to see what would happen. Like, where in the world could these dinosaurs live under certain conditions? Of course, we don't really know a lot, exactly, very precisely, what the metabolism of these animals were like, and it is, of course, a very controversial subject. So they actually modeled several different permutations, you could say, to see how this would impact the survivability of these different dinosaurs. So first, they tested a core temperature range, which you can see is labeled on the left here on these slides. Core temperature range, so how much their body temperature fluctuates. Like, does it fluctuate a lot, like a squamate, so something like a lizard is mostly an ectothermic animal, so it gets its body heat mostly from its environmental temperature. Does it fluctuate a lot? Is it so that would be a high core temperature range. Is it a mid-core temperature range, like a monitoring? So things like echidnas and platypuses, echidnas especially, I think, they do generate their own body heat, but they don't kind of keep their body temperature stable at a certain point like we do. So their temperature actually fluctuates a lot even when they're active. So kind of a mid-core temperature range. In the low core temperature range or narrow core temperature ranges would be something like us. Or in this case, they modeled it based on a bird. So something that keeps its body temperature very stable at a given point. They also modeled different rates of metabolism, so resting metabolic rate, which is labeled RMR on here. So again, they use lizard, monotremes, and bird as different models for this. So lizard has a relatively low metabolic rate, monotreme is a moderate one, and bird generally has a very high one. So the different conditions. And lastly, for Coelophysis, they didn't do this for Plateosaurus because sauropodomorphs so far have not been found with evidence of feathers or filamentous body coverings. But we do know they were very widespread in theropods, although we don't know whether the earliest theropods had feathers or not. But they decided to model kind of different feather coverings for Coelophysis. So either without feathers or partially covered with feathers, like only the back was covered, or kind of full body covering of feathers. All right, so I guess Plateosaurus is a slightly simpler model here, so let's talk about it first. So what they found with Plateosaurus is that under a narrow core temperature range, so it's kind of something similar to us, and under relatively high, like either moderate or high metabolic rates, would give Plateosaurus the greatest range of survivability in the Triassic climate. So, which seems to support that Mesozoic dinosaurs had some kind of elevated metabolism of some sort, and that they weren't lizard-like, operating at lizard-like metabolisms. However, especially at very high metabolisms, high metabolisms, Plateosaurus would have found it quite difficult to survive in very hot regions during the Triassic, so places close to the equator, because it was such a large animal that if it had a high metabolic rate, that it probably wouldn't have been very comfortable. Would have been quite heat stressed, which is quite interesting because these sauropodomorphs, these early sauropodomorphs, are quite common in some places during the Triassic. Their fossils are commonly found, but so far they have not really been found near the equator. So that is a very interesting consistency with what we would expect to see if these animals had relatively high metabolisms. So that I guess that's kind of the main takeaway for Plateosaurus. At least that's what I got out of reading the paper. Whereas for Coelophysis, what they found was that Coelophysis could tolerate a greater range of temperatures if it was covered in feathers. So it could survive in warm environments or cold environments, no problem. However, if it had a full body covering of feathers, it would not have been very comfortable in the kind of warmest regions during the Triassic. But we do know that we do have Coelophysis fossils found, in places that would have been close to the equator during the Triassic, which perhaps suggests that, maybe Coelophysis did not have a full body covering of feathers, although having some would have nonetheless been beneficial. In fact, the fully unfeathered Coelophysis is almost like completely non-viable in any kind of Triassic climate, except in in the very hottest regions under a low core temperature range, so very stable temperature range. Um, so might this suggest that Coelophysis would not had feathers but was not fully covered in them, perhaps? We do also have Coelophysoids, so similar dinosaurs to Coelophysis, found in places that would have been relatively cool during the Triassic. So it could have been that Coelophysoids were quite variable in their body covering. So some of them were partially feathered, some of them were fully feathered, and certainly we see this kind of pattern in modern animals too. So mammals can be quite variable in the amount of body covering that they have, even among closely related species. Now, this study does require a lot of assumptions to go into it. So, obviously, like I described, that there are so many environmental parameters and the parameters regarding the biology of these animals that, you know, it is very difficult to say, like, how solid some of these parameters are when you enter them into this program. But it is a very interesting approach to studying the biology and of these dinosaurs, and it does seem consistent with what at least some people have suggested in the past, that dinosaurs have elevated metabolic rates, and potentially even the earliest dinosaurs could have been feathered. So these are these results are consistent with that. So I would definitely be very interested in seeing this method applied to other extinct animals. And from what I've heard, that seems to be what this team is working on. So I look forward to seeing some of that.
Yeah, that's exciting.
Yeah. Do you have anything else to add to that?
No, I'm good.
Okay, sounds good. We can get on to your stories then.
Yes, absolutely. So I found two from widely different fields, but I thought that they were particularly fascinating. So this first one here, this is a study that, long story short, it provides support for a phylogenetic relationship between the myriapods, which is a lineage of arthropods that includes the centipedes, and the millipedes, and their kin, these multi-legged terrestrial organisms, and a poorly known group of aquatic arthropods called euthycarcinoids, and I hope I'm pronouncing that right.
Yeah, I think you are.
These arthropods come from the Middle Cambrian period and seem to go extinct in the Middle Triassic period, so they don't appear to be casualties of the infamous Late Triassic extinction event. Of course, that remains to be seen. Now, prior to this study, there was a proposed relationship to the mandibulates. So it was at least tossed around that euthycarcinoids had something to do with not only myriapods but also crustaceans and by extension insects, because they evolved from within the crustacean radiation.
Right.
But there was a study in 2018 by Vanier et al. That actually allied this group to the myriapods specifically. So there was some discussion as to whether this lineage should be allied to the living myriapods or not. And so for this particular paper, they analyzed fossils belonging to just one species, and it's the one listed here. This is Heterocrania rhyniensis, and if the species name rings a bell, that's because it derived from the early Devonian rocks of the Rhynichert from Scotland. This is a famous site, which would have been a tropical freshwater bog in life. So this paper acts twofold as a phylogenetic analysis as well as a descriptor of these brand new fossils, because we've known about this organism before, but some recent finds were found that were much more complete and much more detailed in terms of the anatomy that they preserved. So they were able to kind of clarify a few things that were previously unclear. And it was noted that there were many anatomical features that allied Heterocrania, and thus the euthycarcinoids in general, to the myriapods. And I have three of them here. For one thing, there are the aspects of the preoral chamber and the hypopharynx. For those of you not in the know of arthropod anatomy, the preoral chamber is basically the region in front of the mouth where the mandibles lay. And the hypopharynx is found behind the mandibles, and it's sort of where the saliva works to process food. And so if you're thinking about vertebrates like us, then that area corresponds to the area in our throat where our voice box lies. So arthropod mouths are pretty weird. Of course, you could flip that and say that vertebrate mouths are pretty weird.
Right.
But we both find ways to eat food, so it kind of works out.
Right.
So there are similarities with those features with the myriapods, but as well as the compound eyes. So you might be familiar with the eyes of houseflies or other organisms, these really beautifully intricate structures. For one thing, these new fossils of Heterocrania confirm the existence of these types of eyes for euthycarcinoids in the first place. because the previous remains they're very poorly preserved. We couldn't tell for sure what sorts of eyes that they had as far as arthropods go. Incidentally, compound eyes are plesiomorphic traits. They are found among ancestral arthropods as well as the stem lineages before them. And the fact that this type of eye is not only occurring among euthycarcinoids, but also some of the earliest diverging kinds of myriapods. So there's a lineage of centipedes and a lineage of millipedes where they retain the compound eye, but all the rest have modified these to simpler structures with just a simple lens. So this sort of provides an anatomical link between this plesiomorphic sequence, and this has been used to argue that euthycarcinoids are not only related to myriapods, but they're stem myriapods. So there's been a lot of talk recently about crown groups versus stem groups. Crown groups are basically the common ancestors of all the living members of a lineage. So crown group myriapods would be the centipedes and the millipedes. But stem groups diverge before the crown group. They're typically extinct, but not always.
They're always extinct by definition.
Wait, I'm sorry?
They're always extinct by definition. Yeah, sorry.
Oh, really? Oh, I'm sorry. Yeah, Albert's right. So, for example, stem birds would be things like tyrannosaurs and sauropods, technically.
Right.
Whereas crown birds would be, as previously discussed, ratites and galloanceriforms and whatnot. So euthycarcinoids would take the stem lineage to myriapods. Albert, if you could change the slide for me.
Sure. Let's see. Yep.
So here I've whipped up a simple phylogeny of arthropods, and you can see the placement of the euthycarcinoids as a sister group or stem lineage to the myriapods. So once this was established through anatomy, the authors turned to geologic and molecular data to kind of help cement this decision. There is a set of trackways attributed to an Ordovician euthycarcinoid of different species, and they seem to show an animal that's using a millipede-like locomotion along a tidal environment. And so that's particularly fascinating because the euthycarcinoids are found in marine and freshwater deposits, whereas myriapods, for the most part, are terrestrial. Albert, you brought to my attention that there is at least one known aquatic centipede that's alive today, and incidentally, that was a recently described species. So for the longest time, we had the understanding that myriapods were totally terrestrial. So this sort of provides a possible mode of transition between an aquatic existence and a terrestrial one that gave rise to the myriapods, which had already been expected. The ancestors of the crustaceans were aquatic. The ancestors of the chelicerates were aquatic. So it seemed reasonable that the ancestors of the myriapods would be aquatic too. And so they looked at the molecular clock and they analyzed that the myriapods seem to have diverged from the other arthropods between the end Ediacaran and the Cambrian period. And since the earliest euthycarcinoids are mid-Cambrian age, that's been used to kind of lock these two groups together in support of this stem relationship, which is effectively bridges the wide gap in the geologic record between the earliest fossil myriapods and their ancestors as proposed by the molecular clock. So I thought that was particularly fascinating. Don't see a lot of press usually on the myriapods. Albert, do you have anything you wanted to
add? Sure. I'll briefly say. So a lot of you might not know this, but I did my master's project looking at crustacean phylogeny and crustacean evolution while I was doing a paleobiology degree in the University of Bristol. And while I was doing that, basically I was doing a combined analysis using morphological data and molecular data together, including morphological data from fossils, and I was using a data set that had originally found the euthycarcinoids as a group of crustacean. And so I originally included the euthycarcinoids in there, but one of my supervisors, who was actually the one who made that matrix, that data set, he told me, oh, no, there are studies coming out that show that euthycarcinoids are stem myriapods, so don't bother including them. So, yeah. Right. Anyways, that's just a fun side anecdote. But other than that, we do have we have roughly 10 minutes left for kind of discussion, so we should probably continue onward to your next story.
Sure, and I'll try to breeze through this as quick as possible because this one particularly caught my attention.
Yeah, I know. You have a lot to say, but yeah.
Yeah. So, okay, this study This essentially further fleshes out the story of the peopling of the Caribbean islands. This is one of the last regions in the Americas that were settled by human beings, and also is it's the launching point for the Columbian Exchange. So it's a particularly important area of research in anthropology. And Albert, if you could change the slide for me,
Yep.
please. So here I have two diagrams. The one on the left comes from this particular study. This shows basically the chronology and the DNA samples that were used. And the one on the right is one that I mocked up. This is essentially the state of knowledge for Caribbean archaeology. So you're looking at different technological sequences. So previous studies demonstrated multiple migrations of people entering the Caribbean over many thousands of years. And in archaeological context, these two periods have been given the names Archaic and Ceramic in terms of the pre-Columbian history. And so just to clarify briefly, archaic groups are those that are working with permanent settlements and incipient agriculture. They're sort of on the transition from hunter-gatherer societies to agricultural ones.
Mm.
And while the name might suggest otherwise, there is some evidence that they may have been making pottery. Now, this is in contrast to these ceramic groups, which certainly were agriculturalists, and they almost certainly used pottery, some of which was very highly detailed. And it's from these ceramic groups that we get the later contact cultures, the Taino nations and the island Caribs. And so in archaeological literature, these have been further subdivided, perhaps controversially, into several different cultures, some of which I'll be mentioning here, based upon the specific features of the technological remains, particularly the pottery. So with this particular study, it's addressing a number of things, perhaps the three biggest issues in Caribbean archaeology today. For one, it's the actual chronology of settlement, trying to figure out when specifically groups started entering this region. Two, the actual routes taken from the mainland. And three, the nature of interactions between these different cultural groups. So was there admixture, was there elimination, or anything in between? So Albert, if you could change the slide for me, please.
Yes.
So this is another figure from the paper. I'm sure I might be butchering this name. Nagel et al. They attempt to answer some of these questions by running an ancient DNA analysis by charting these migration routes through a phylogenetic method. So the baseline data set includes the DNA of 93 individuals from 16 different sites all across the Caribbean, dating from about 3,200 to 400 years ago. The archaic data set comes specifically from 7 Cuban sites, while the ceramic data set comes from across the different islands. And I should specify between the Greater and the Lesser Antilles. The Greater Antilles are essentially Cuba, Hispaniola, Jamaica, and Puerto Rico. And the Lesser Antilles are all of the other little islands that bridge the Caribbean Sea towards the South American continent. And this is alongside select genetic data from across the Americas for use as outgroups, as you can see here.
Mm.
Now, the consensus model prior to this study shows that the earliest archaic Caribbeans inhabited Cuba and Hispaniola, and they seemingly arrived from a Central American route around 8,000 years ago. And then there were later migrations of archaic Caribbeans going into the Lesser Antilles, possibly from a South American route for about 5,000 years ago. And then later on, these ceramic groups all seem to have migrated from South America specifically. And this would have been about from 2,800 years onwards. And they would have settled first in Puerto Rico and the Lesser Antilles, and then eventually spread westward into the Greater Antilles. And so there is a big debate as to whether these ceramic groups were island hopping across the Lesser Antilles or directly jumping from the mainland of South America to the Greater Antilles. And so, Albert, if you could change the slide for me again, please. This is a figure that I mocked up, kind of summarizing the data here. So Nagel et al. Seems to confirm these models while adding some new details, specifying at least 3 separate movements into the Caribbean, 2 archaic and 1 ceramic. So for instance, the archaic migrations do not appear to have belonged from the same source. This first migration would have been tied to the very first widespread movements of the earliest Amerindians from North America, which would have been before the greater settlement of the South American continent. And this would have been at least after 11,000 years ago or so. With the other archaic migration arriving in the Caribbean thousands of years afterwards. This is derived from a previous study of Posh et al. 2018, which, long story short, provides evidence of a very complex genetic history for both continents, including continent-wide gene flow and just absolute waves of population replacements. It's a very fascinating story that's slowly being uncovered. One of my biggest issues with this paper, before I move forward, is that there tends to be this issue among DNA researchers, ancient DNA researchers in particular, and this is not a value judgment at all on the author's concern. There's been worries from archaeologists who have previously worked on this using, sample sites, bioanthropology, linguistics, and so forth, that these ancient DNA researchers are not consulting previous work when they develop new studies. So when they uncover these results from DNA, they're revealing research that seems brand spanking new and surprising,
Right.
but it's already been studied in the literature, and oftentimes there's absolutely no credit given from these previous researchers, effectively eliminating their voices from the conversation. In particular, there was a recent study a couple of years ago looking at the population history of ancient Egypt, and this particular team of ancient DNA researchers found a general population continuity before the Roman period. You start seeing influences from Europe as well as sub-Saharan Africa. And of course, the researchers were like, "This is amazing! This is incredible!" But then you have all these Egyptologists coming in saying, "What, you just now figured this out? We knew about this for years from studying the bones and the different sites."
Right.
And so I've noticed quite a lot of that here. So for this presentation, I have highlighted and included aspects of Caribbean archaeology that were otherwise missed in the study to kind of help tie these things together and maybe provide some context for some things. So just to flesh it out a little bit, this first archaic migration might be tied to what is known as the Casimiroid culture in the archaeological literature, which is known as one of the first groups of people living in the Caribbean. And there are ties from remains in Belize, which suggest a possible amount of road migration. Now, the paper itself here does not recover any information about launching points to the Greater Antilles. So perhaps if they had used data from Belize and neighboring sites, they might help clarify some of the things that they were asking. Now, for the later archaic migration, and this is an interesting observation, the authors state that the DNA requires some South American-related ancestry to make their model fit. So there's about 39% of South American ancestry within the individual studied here, versus a larger percentage of Central American-related ancestry, so about 61%. Now, we know from the archaeological literature that some of the earliest sites in the Lesser Antilles and westward derives from a culture known as the Ortoroids, or the Ortiroids. That's a tough one for me. And then, just like in the Central American study, these have been tied to sites in the Guianas, so further in the Brazilian region. So it's possible that this South American ancestry might derive from there, but it is an open question as to what all of this Central American data means. If there was maybe groups moved from Central America into South America before moving, or maybe there was a meeting up at some point. It's an open question. And so, Albert, if you could turn to my last slide, please, as I wrap this up here. So regarding these ceramic data, at least one migration was detected in the study, originating from South America. And this would have been about after 2,000 years ago. Now, again, this is not in the paper at all, but this date allies very well with the proposed Saladoid migration, which would have occurred at the same time, 2,500, 1,400 years ago. And this would have been one of the first of these ceramic period movements. Now, this is a curious thing because the archaeological evidence has suggested there were at least 3 movements from South America for the ceramic groups. And if you read the literature, these have been proposed to have been derived not only from the Saladoid center in the Orinoco region, as I have listed here, but they also are extensions of the Saladoid societies, and they vastly changed the cultural influences of these later Caribbean groups. So the fact that there doesn't seem to be any trace of this in the genetic data is very interesting indeed. Now, the authors state that this island hopping model that I previously proposed best fits the data, but they don't say how. They just kind of leave it at that, adding that they admit that they need more genetic data from more sites to really make this model more widely known, because they only used 2 islands in the entire analysis from the Lesser Antilles. If you saw in the previous slide with the 3 maps, there's a phylogeny branching from St. Lucia to Puerto Rico and westward. And so I think that's what they're maybe getting this from. Now, if you look at the literature, you'll notice that the island hoppers and the direct jumpers bring up very good points. Previous work has shown that trade winds and ocean currents, along with easy visibility between these islands, makes island hopping a very reliable way of colonizing the Caribbean. In fact, it only takes a couple days to move between some of these islands. It's not like the Austronesians, that is the ancestors of the Polynesians. while they have to rely on outside factors in order to see the islands that they're trying to get to. They couldn't actually see as they were moving, whereas these island hoppers would have supposedly done so. But the direct jump route is just as viable. The earliest ceramic sites in the entire Caribbean are found on the Leeward Islands and westward, so at the very northern part of the Lesser Antilles. So, when you look at the archaeological literature of the contact period, when you look at the writings of Columbus, for example, you see that these indigenous boats that were used, these are called skegs, these would have been more than capable of making these large sea voyages across the Caribbean Sea to get to, say, Puerto Rico, without having to worry about island hopping to get there. Now, it is highly possible that settlement was a lot more complicated than this sort of simple dichotomy between just island hopping and just direct jumping. I mean, it's easy to think that maybe they started island hopping and then made a direct jump, or maybe they made a direct jump and then just island hopped downwards, going back into South America. It's interesting to think about. Now, just wrapping up here, there was a brief discussion, but not major, regarding the possible cultural contacts between the archaic and ceramic groups. Because if you look at the archaeological evidence, there is a major stalling of about 1,000 years of ceramic groups in the Caribbean before they start moving into the Greater Antilles and overtaking the archaic groups. So that suggests that there were at least some social barriers to movement, but what kind, we have no idea. When looking at the DNA evidence itself from this study, they only find admixture between archaic and ceramic groups with one individual from Puerto Rico. This would have been about 1,000 years ago or so. So the authors were very surprised about this, and if you look at the archaeological literature, it is very surprising because you see nothing but waves and influences between these different cultures. So again, this might be just an artifact of limited sampling. So in conclusion, the authors state, and I quote, "There is a mounting evidence to suggest that far from being an insuperable barrier, the Caribbean Sea functioned as an aquatic motorway that people crossed frequently despite its occasional unpredictability." So there certainly is much more work that needs to be done in unraveling this lost history, but it's very clear that it's much more simple than you might suspect.
Yep. Sounds good.
Albert, did you have anything you wanted to comment on?
Oh,
I know
well,
we're a little booked for time.
it's okay. I don't think we received any questions in the meantime anyway, so that's fine. Um, I don't think there's anything I could add to that is particularly intelligent, but it's a fascinating topic, absolutely, the topic of human migrations and how they happened, and especially a complex example like this. So that's all the stories we had for you today. Hope you enjoyed the show and continue to enjoy the rest of the stream.
Yes, thank you so much for listening to us.
Yeah,
And thank you so much for your donations, too. We really appreciate it.
right. So when we reconvene, I'll stop the stream shortly. When we reconvene, I will be back with our next guest, which will be Gabe Santos from the Raymond Alf Museum in California. See you in a bit.
Take care, everybody.